S Kitty here, and you're in for another WTF Science. Today we'll be talking about mitosis, or mitotic cell division. In order for a cell to divide into two, i.e. two daughter cells, a cell must go through mitosis. Now, in order for a cell to get to mitosis, a cell must first go through interphase. An interphase is actually a combination of different phases called the G1 phase, the S phase, and the G2 phase. Now the G and the G phases stands for gap. There are two main parts of the G1 phase. First, the cell must have growth, and second, the cell must survey its environment to decide whether or not it should divide or not. If division conditions are not optimal, the cell will enter a sort of hibernation, sometimes called the G0 phase. The next part of interphase is the S phase. Now the S for S phase stands for synthesis, and this is where all the DNA replication occurs. Our cells inside the nucleus, our entire DNA or a genome is kept, all of it, and it's all rolled up into this nucleus. And so in order for a cell to properly divide into two functioning cells, there must be a copy of DNA in both of them. Somatic cells, before they go through DNA replication, have two sets of 23 chromosomes that make up the 46 chromosomes we have. I'm going to show you some helpful images to show you what happens in S phase, but keeping in mind that I'm only showing two, one each, a chromosome from each parent that we get, as opposed to the 23 from each parent we get. So it's not like, keep in mind that there's actually 23 chromosomes going through this on each side. So first there's the cell before DNA replication. This consists of a chromosome from mom and a chromosome from dad. These are called homologous chromosomes. Now after the chromosomes are replicated, they become chromatids, and when there's a set of two of them, they're called sister chromatids. So you have your dad's, the sister chromatids from your dad's side and the ones from your mom's side. Now these are attached by something called the centromere, and it's also called the kinetochore is also there, but we'll get into that later. The next phase after S phase is called G2 phase. Now G2 phase is also a phase of growth, but it also in this phase of growth, the cell grows components and makes components that it will need through cell division. This is also an important checkpoint in cell division. Now what a checkpoint is it's this point during cell division where the cell checks to make sure everything's going properly. This is in order to try to avoid mutations or diseases that can come from mutations because they can be caused by the cell not dividing properly. There are a few checkpoints throughout mitosis. At the end of G2 phase is one of the major ones, and right before a cell goes into anaphase, which we'll get to in a bit, is another one. So after the cell makes sure that all the DNA has been replicated properly, it can now start going through mitosis. Now the first stage is prophase. The chromosomes in the cell condense, and the nuclear envelope begins breaking down. The next phase is prometaphase. The nuclear envelope is now completely fragmented, and the mitotic spindle is fully formed. The microtubules that originate from the mitotic spindle attach to the centromere or kinetochore of the chromosomes. In metaphase, the chromosomes line up in the exact center of the cell. They are equal distance from the poles of the mitotic spindles. Following metaphase is anaphase. Now this is when the chromosomes start to separate and the cell starts to push apart. Now the overlapping microtubules help push the cell apart, and when they're the ones connected to the kinetochore help pull the chromosomes to each pole. Finally, the cell enters telophase. Now, this is when cytokinesis is probably occurring, the separation of the cytoplasm and the reformation of the plasma membrane. Then you will have two cells, and these will loop back around and start at interphase all over again. So I just wanted to talk about the mitotic spindle for a little bit so you can like understand what it is. Now, the mitotic spindle has centrosomes. There are 3 different types of microtubules, and they all originate from the centrosome. Now, there are the polar microtubules, which are the ones that overlap and help push the cell apart. Aster microtubules, and those are the short ones, and they kind of help position the centrosome. And finally, there's the kinetochore microtubules, which are the ones that attach to the kinetochore and pull the chromosomes apart. The centrosome, which the microtubules originate from, should not be confused with the centromere, which is the center of these chromosomes. And that's how mitosis works. Be sure to go check out WTF History. You got Grindle there with a great blog, and be sure to comment, subscribe, and follow on Tumblr. I am always reading the comments that are posted, always reading my messages on Tumblr, even if I don't respond. But thank you for watching. I hope you enjoyed, and I hope you learned something, and tune in 2 weeks.